All right, so we're going to talk about enzymes here. So an enzyme is a type of protein. Therefore, it's made up of the amino acid monomer. And so enzymes have special jobs. But before we talk about enzymes, first thing we have to understand is activation energy. So in any chemical reaction, you need, in order for the reaction to proceed, you need a certain amount of energy. So it's the amount of energy needed for reactants to form products. And so sometimes the activation energy can be really high, and sometimes there is a very low activation energy, which means the reaction happens pretty quickly. So in your body, most things have high activation energy. And so you don't have a lot of spontaneous chemical reactions occurring. And so we need something to speed up these reactions, because a lot of these reactions are important for life, right? And so in your body, there are catalysts. And in general, a catalyst is a substance that lowers the activation energy. What it does is it speeds the reaction up. In your body, there's a specific type of catalyst called an enzyme. As I said before, these are made up of proteins, so enzyme is a protein. What they do is they're a catalyst, so catalysts speed up reactions. They speed up the rate of the chemical reaction by lowering activation energy. So if we were to recap really quickly, every chemical reaction requires energy, and that's called an activation energy. In your body, a lot of chemical reactions require have high activation energy, so we need something to speed it up. In general, a general term for something that speeds up a chemical reaction is called a catalyst. But in your body, there are specific catalysts called enzymes. And they're a type of catalyst, so they speed up the reaction by lowering the activation energy. How do enzymes work? Well, first of all, an enzyme, like any catalyst, they're not used up in reactions. So they're not really part of the reaction, they just help in speeding up the reaction. They can be used over and again, and most enzymes are specific to one reaction. So there's an enzyme for each chemical reaction, and the reason why is it's specific to the substrate, and we'll talk about what a substrate is. The big thing here is we want to know that most enzymes end in ASE. So every time you see ASE, usually that refers to an enzyme. So one of the most common ones is lactase. So lactase breaks down lactose, which is found in dairy products. So a lot of people are lactose intolerant, and the major reason is they either have no lactase, so they don't have the enzyme to break down lactose, or the lactase is mutated somehow. Okay, so what exactly happens, or how does an enzyme work? So let's just say I have an enzyme, and I'm just going to draw an enzyme kind of looking like a Pac-Man. So here's my enzyme. So the enzyme has a specific shape, and that shape usually is dependent on what they're going to bind to. So for example, this enzyme, let's say it's going to bind to, it looks like a triangle, right? And so this is called the substrate. The substrate is what the enzyme binds to, and so this is the reactant. Well, where does the enzyme bind the substrate? It binds it in what we call the active site. So this is called the active site. All these terms you need to know. So the enzyme finds, now there's different types of substrates, right? Like there might be a substrate that looks like a box here. And so this is a different type of substrate. What I want you to notice is the enzyme, this substrate will not fit with this enzyme. So maybe for this substrate, there's a different enzyme that has active site that fits the substrate. So what happens is the enzyme here will bind the substrate, and then what happens at this point is the enzyme kind of speeds up the reaction. So let's say the reactant was we had this, and it's going to break down into smaller triangles. So our products is 2 smaller triangles, right? And so what happens is once it's in the enzyme in the active site, it maybe breaks it apart so that once it breaks apart, the enzyme will release, and then you have now 2 smaller pieces. And these are your products. So what the enzyme did basically was it kind of facilitated the reaction by binding the substrate. And so it kind of sped it up. Maybe over time the substrate will produce this product, but this takes a long time. So this reaction takes a long time. So the enzyme, what it did is by binding it, it actually sped up the process. It overcame or lowered what we call the activation energy. So there are things that affect enzyme action, and so enzymes, if you heat up the temperature, or you increase the temperature, oftentimes it'll start speeding it up. But at some point, the proteins actually start breaking down. We call denature. If it's too cold, the enzymes usually don't work as fast, and so it slows it down. So temperature is one. pH is another. Each enzyme has a specific pH that they work in. And the last one is the concentration. So the more of the substrate, the more reactants, the more likely that the enzyme will run into the reactant. And so therefore, it will help speed up the reaction. Let's just do a quick example. So it says, what is the optimal temperature for this enzyme? Remember, this here is the rate of the reaction. So the highest rate of reaction is right here, right? So it's at this temperature, say around 40 degrees Celsius. And so you'll notice is that if it gets too cold, like over here, the rate of reaction is low. And if it gets too hot, well, the protein starts breaking down, the enzyme starts breaking down, and so the rate of reaction lowers. So there's like a happy middle. The same thing occurs for pH. So in pH, here are 3 types of enzymes. And so you'll notice that the question is chymotrypsin. Where does it function best? Well, here's the enzyme activity. So here's the highest point for, and you'll see it functions best at 7. So you'll see it functions around pH 7 or neutral, whereas say trypsin right here functions best at like 9.5. Pepsin's maybe at 2. Okay, and so we have to be able to read these graphs in order to see how they function. But to recap, we just want to go over that there are 3 things that affect enzyme activity, or how well the enzyme works. One is temperature, another one is pH, and then the last one is the concentration of salt of the substrate.